Picture this. It is 3 a.m. You are still awake, and the work is still in front of you. Your eyes feel like they have been packed with sand. You reach for your coffee, and it takes a second too long for your hand to actually move. You read the same sentence 3 times, and it still does not land. Your working memory, the mental whiteboard you use to hold an idea while you work with it, has basically gone offline. You are not just tired. Something has gone genuinely wrong inside your brain cells, and it started hours ago, quietly, without asking your permission. Most people assume that feeling is just what happens when you skip sleep. A willpower problem, a discipline problem, something coffee fixes, or at least masks. But what is actually happening at that moment is a specific energy crisis inside your neurons. The machinery that keeps your thinking sharp runs on a fuel called ATP, and your brain has almost no reserve tank. When the demand for that fuel spikes and the resupply slows, your cognitive performance does not gradually fade the way a dimmer switch works. It drops off a cliff. Here is where creatine enters the picture, and here is where most people have the story completely wrong. Creatine is a muscle supplement. You take 3 to 5 grams a day. Your muscles load up, you lift more, you recover faster. Simple. And if it has some vague brain benefit somewhere in the fine print, great. But that is the bonus, not the point. That is the version almost everyone believes, and it is incomplete in a way that actually matters, because the brain is not muscle. The wall surrounding your brain controls exactly what gets in and how fast, and creatine does not cross it the way it floods into skeletal muscle. The dosing logic that works for your biceps does not transfer to your neurons. And the emerging research is not about daily low-dose supplementation for general focus. It is about something far more specific, a higher acute dose taken before a predicted window of sleep deprivation, and the cognitive signal it may produce when the brain's own energy reserves are already running thin. Today we are going to break this down mechanism by mechanism. We will start with why the brain's energy system is so different from muscle, then trace exactly what sleep deprivation does to that system at the cellular level, and then we will examine the 2 acute dosing signals the research has actually tested, what approximately 0.2 grams per kilogram and 0.35 grams per kilogram appear to do, what they do not do, and where the evidence genuinely ends. By the time we are done, you will understand why metabolic state and timing may matter more than daily gram counts, and you will be able to evaluate every future claim about creatine and brain performance with a clear framework instead of a guess. Let's start with the brain's energy problem, because it is more extreme than you probably think. So, let's start with what is actually happening inside your skull when you are running on no sleep. Not the feeling of it, the machinery underneath it. Your neurons are some of the most energy-hungry cells in your entire body. The brain makes up roughly 2% of your body weight, but it burns closer to 20% of your total energy supply at rest. Think about that. One organ, 20% of the fuel, and it is running that tab constantly, whether you are solving a math problem or staring at a wall. The currency it spends is ATP, adenosine triphosphate. Every time a neuron fires, every time it passes a signal to the next cell, every time your working memory holds a piece of information in place while you do something with it, ATP gets consumed. It does not get stockpiled in any meaningful quantity inside neurons. Your brain cells are not like a camel storing water for the desert. They are more like a city with no reservoir, fed entirely by a continuous water main. The moment that flow slows down, the taps run dry and the lights start flickering. This is where phosphocreatine enters the picture, and it is genuinely one of the more elegant pieces of cellular machinery you have running inside you right now. Phosphocreatine is a rapid response buffer. When ATP demand spikes suddenly, faster than the slower metabolic pathways can respond, phosphocreatine steps in and donates a phosphate group to ADP, which is the spent form of ATP, and regenerates ATP almost instantly. Think of it like a reserve generator. The power grid is your main metabolism. But when demand outstrips supply for even a few seconds, the reserve generator kicks on and buys time. Phosphocreatine is that generator. Here is the part most people do not know. Your brain does not just import all its creatine from food and supplements. It actually synthesizes some of its own, locally inside brain tissue. The brain has its own creatine production line, That is a remarkable thing because it tells you something important. The brain treats creatine as critical enough to make its own supply rather than relying entirely on whatever comes in from the bloodstream. And it also helps explain why the brain does not respond to external creatine the same way your muscles do. It has its own system, its own rules. And between that system and your bloodstream sits one of the most sophisticated security checkpoints in biology, the blood-brain barrier. You have probably heard the name. Most people picture it as a kind of filter, something that blocks toxins and drugs from reaching the brain. That is roughly right, but the engineering behind it is worth understanding because it completely changes how you have to think about creatine reaching your neurons. The barrier is not a membrane in the traditional sense. It is a layer of endothelial cells, the cells that line blood vessels, that are joined together so tightly there are virtually no gaps between them. In most of your body, capillary walls have small openings that allow molecules to pass in and out fairly freely. In brain capillaries, those gaps are sealed shut. Surrounding those endothelial cells are astrocytes and parasite cells that add another layer of control. The result is a wall that forces almost everything to pass through the cells themselves, one molecule at a time, through specific transport channels built for the purpose. Creatine crosses this barrier through a transporter called SLC6A8. That transporter is specific to creatine. It actively pulls it from the blood into brain tissue, and it has a ceiling. It saturates. Once the transporter is working at full capacity, pouring more creatine into your bloodstream does not open a second lane. There is no second lane. The transporter is the only gate, and it can only move so much creatine at a time. This is where the entire muscle supplement logic breaks down when you try to apply it to the brain. In the standard muscle loading protocol, you take around 20 grams of creatine per day for 5 to 7 days, and you rapidly saturate your muscle creatine stores. That works beautifully for muscle because skeletal muscle has high transporter capacity and a strong gradient that pulls creatine in. Researchers who have actually measured brain creatine using a technique called magnetic resonance spectroscopy, which lets you quantify specific molecules inside living brain tissue without cutting anything open, have found that brain creatine does increase with sustained supplementation. But the increase is modest compared to what happens in muscle. The brain's transporter is the rate-limiting gate, and it does not swing open just because there is more creatine available in the blood. So, if you have been taking 5 grams of creatine a day thinking you are loading your brain the same way you are loading your muscles, the evidence suggests the picture is more complicated than that. The muscle is responding. The brain, protected behind its barrier and its saturatable transporter, may be doing far less with what you are sending it. Now, layer onto this what actually happens when you stop sleeping. Extended wakefulness is not just fatigue. It is a measurable metabolic stress event inside your brain. One of the clearest chemical signals is adenosine. As your neurons fire throughout the day, adenosine accumulates as a byproduct of ATP consumption. It builds up progressively, binds to receptors in the brain, and signals that the system is tired and needs rest. This is incidentally exactly what caffeine targets. It blocks adenosine receptors without clearing the adenosine itself, which is why the crash returns when the caffeine wears off. The adenosine was always there, waiting. But beyond adenosine signaling, sleep deprivation shifts cerebral glucose metabolism in ways that are genuinely concerning. Some brain regions do not simply slow down under sleep deprivation. They become less efficient at using the fuel they do receive. The neurons are still demanding energy, but the machinery converting that energy into usable ATP becomes less productive. You are burning fuel and getting less output per unit. It is like an engine running on a degraded mixture, consuming the same amount of gas but generating less power. The cognitive functions that fall first are not the most basic ones. Your ability to walk, to recognize faces, to understand language, these stay relatively intact for a long time. What collapses first are the functions seated in the prefrontal cortex: working memory, sustained attention, reaction time, and executive control. The capacity to hold multiple things in mind simultaneously and act on them flexibly. These are also, not coincidentally, the most energy-intensive cognitive functions you perform. They are first in line for the fuel and first to fail when the fuel supply is compromised. Here's the piece that genuinely changes the risk calculation. When researchers measure objective cognitive performance in sleep-deprived subjects, and then ask those same subjects how impaired they feel, there is a striking disconnect. Subjective sleepiness ratings lag behind objective performance collapse. After 17 to 19 hours of continuous wakefulness, measurable performance decrements are already present. After 24 hours, they worsen non-linearly, meaning the decline accelerates rather than continuing at a steady rate. But the person inside that experience often feels less impaired than they actually measure. You are not a reliable judge of your own cognitive deterioration when you are the one being deprived of sleep. The gauge is broken exactly when you need it most. And through all of this, the phosphocreatine buffer in your brain is under pressure. Sustained high cognitive demand under conditions of reduced metabolic efficiency may partially deplete that reserve generator. The buffer that normally lets your neurons buy a few extra seconds of ATP during a demand spike has less in reserve. The system that was already running thin is now also running without its emergency backup at full capacity. This is where the creatine story becomes genuinely interesting. If the phosphocreatine buffer is more depleted than usual because of extended wakefulness, that means there is more room in that buffer to absorb additional creatine and contribute meaningfully to ATP resynthesis. Think of it this way. Imagine a backup battery in your phone that normally sits at 80% charge. When it is at 80%, plugging in a small charger barely registers. But if that battery has been drained down to 20%, the same charger suddenly makes a real difference. The buffer has actual capacity to fill. This is the proposed mechanism for why creatine might matter more during sleep deprivation than on a normal rested day. It is not that creatine becomes more potent, it is that there is more room for it to work. A well-rested brain with a fully charged phosphocreatine reserve has less marginal need for additional buffering capacity. That predicts something important. If you are rested and well-nourished, extra creatine may not move the needle cognitively at all. The evidence from controlled trials in rested individuals generally supports this. Cognitive benefits from standard creatine supplementation in healthy, well-rested, well-nourished people are minimal at best. The state of the system determines the value of the supplement. That is the conceptual pivot this entire video has been building toward, and it has a direct implication for dosing. If reaching a meaningful transient elevation in brain creatine availability requires fighting against a saturatable transporter and a selective barrier, and if that elevation only meaningfully helps when the buffer is partially depleted, then the question becomes, what dose is actually large enough to move the needle at the brain level during that specific stress window? Researchers have tested this, and the doses they used are substantially higher than anything in a standard sports nutrition protocol. The most discussed signal comes from a study using approximately 0.35 grams of creatine monohydrate per kilogram of body weight, administered as an acute single dose before or during a sleep deprivation paradigm. For a person weighing 75 kilograms, that is roughly 26 grams of creatine in one sitting. To put that in perspective, if you have been taking creatine for muscle performance, you are probably taking 3 to 5 grams per day. This is 5 to 8 times that amount, taken all at once for an entirely different purpose. What did the research observe? Compared to placebo, subjects who received the creatine dose showed attenuated decline in cognitive performance measures. Reaction time held up better. Working memory tasks degraded less steeply. Mood and fatigue scales showed less deterioration. The word to hold on to there is attenuated. The decline was slowed. The floor was raised somewhat. This was not a full reversal to rested baseline performance. Nobody took 26 grams of creatine and performed as if they had slept 8 hours. That framing would be wrong, and the science does not support it. What the evidence suggests is a partial buffer against the worst of the cognitive cost, not an elimination of it. The evidence base for this specific dose is not large. There is at least one well-designed human study. Independent replication has not yet been established at scale. That matters. A single study, even a well-conducted one, is a signal worth taking seriously and a conclusion worth holding lightly. The mechanism is plausible, the result is interesting, and the next step is more research, not a population-level recommendation. There is also a practical variable the study context does not always foreground. 26 grams of creatine monohydrate in a single dose is a substantial gastrointestinal load. Creatine at high single doses can cause bloating, cramping, and gastrointestinal distress in some people. The controlled setting of a study does not always reflect what that experience looks like in the real world, and it is worth factoring into any practical consideration. The second dose signal, at approximately 0.2 grams per kilogram, gives you another data point. For that same 75 kilogram person, this is about 15 grams in a single sitting, still well above the daily maintenance dose most people use, but lower than the 0.35 mark. The evidence at this level is even more limited, but the comparison between the two doses matters for a specific reason. If a genuine dose-response relationship exists, meaning more creatine produces a proportionally larger cognitive buffer, that would strengthen the entire buffering mechanism hypothesis. It would suggest the effect is real, dose-dependent, and predictable. If the two doses produce similar or inconsistent effects, the picture becomes more complicated and the mechanism less clean. The current evidence does not firmly establish that dose-response pattern yet. It is a question the research has not fully answered, but the architecture of the question is scientifically coherent, and that coherence is itself informative. Now, before this picture starts to feel like a complete roadmap, there is a critical part of the conversation that does not get enough space in most coverage of this topic. Creatine may buffer some of the cognitive cost of acute total sleep deprivation in a specific experimental context. That is not the same as saying it works for all types of brain fog, all forms of fatigue, or all people in all states. Sleep deprivation in a controlled study is a very specific stressor with a very specific metabolic profile. Jet lag, chronic partial sleep restriction, mental burnout, post-illness fatigue, these states have different metabolic signatures. The phosphocreatine buffer depletion dynamic may look very different in those conditions. The evidence does not currently extend cleanly from total sleep deprivation in a lab to the messy, varied ways people actually feel cognitively impaired in daily life. Rested, well-nourished people taking standard creatine doses show minimal to no documented cognitive benefit in controlled trials. That is an important finding in its own right. It means that if you are sleeping adequately, eating well, and your brain's energy system is functioning under normal conditions, your phosphocreatine reserves are likely sufficient. An additional creatine supplementation is probably not generating a meaningful cognitive effect. The effect, if it exists, appears to emerge from the stress state, not from the baseline. Creatine also does not touch the underlying cause of sleep deprivation. It does not reduce adenosine. It does not restore sleep architecture. It does not clear the accumulated sleep debt. If you use a buffer to get through an all-nighter and then do not sleep, the debt remains, and the buffer has done nothing to address it. There is one more layer worth sitting with. Creatine transporters in the brain, like transporters elsewhere in the body, may downregulate with chronic oversupply. This is a pattern observed in muscle tissue. Sustained high creatine exposure can reduce the transporter's expression over time as the cell adapts. Whether this happens in brain tissue under repeated acute high-dose conditions is not yet confirmed in humans, but it is a biologically plausible concern, and it is one reason why positioning this as an occasional situational strategy rather than a repeated high-frequency protocol is the more defensible approach given what the science currently shows. What the practical picture looks like, given all of this, is fairly specific. The metabolic context where this mechanism is most plausible is anticipated total sleep deprivation, a known overnight shift, a transcontinental flight followed by immediate cognitive demands, an extended work sprint that eliminates a night of sleep entirely, not chronic partial sleep restriction, where you are getting 4 or 5 hours a night for weeks. The mechanism being proposed involves an acutely depleted buffer, and that dynamic is most clearly present in total acute deprivation, rather than the slow, grinding energy drain of insufficient sleep over time. Timing relative to the deprivation period appears to matter in the studies that have been examined. This is not a casual take-it-whenever strategy. The form used in all relevant studies is standard creatine monohydrate. There is no evidence that creatine hydrochloride, creatine ethyl ester, or any of the other marketed forms of creatine produce the same effect in this context, because those forms have not been tested in this context. Monohydrate is what the research used, and individual variation is real. Creatine transporter expression differs between people. Some individuals are known as creatine non-responders for muscle purposes, meaning their muscles do not load creatine well, even with standard supplementation. The same variation in transporter biology almost certainly affects brain response. This is not a compound that produces a uniform, predictable effect across all individuals in all states, and anyone presenting it that way is simplifying past the point of accuracy. Here is the reframe that makes the most sense of everything covered so far. Think about how experienced caffeine users actually use caffeine. Not everyone drinks it every morning out of habit. Experienced users often time it strategically, before predicted periods of high demand, saving it for when the need is real rather than habitual. The evidence points toward creatine behaving similarly in this context, most valuable when deployed situationally before a known acute stress event, rather than taken daily as a general cognitive maintenance dose. That is a fundamentally different relationship with the compound than the one most people currently have with it. So here is where you actually are now versus where you started. You came in thinking creatine is a muscle supplement with some vague bonus for your brain if you remember to take it every morning. You leave knowing the brain is structurally walled off from easy creatine access, that the transporter controlling the gate saturates, that the muscle loading model does not transfer, and that the research pointing toward a real cognitive signal is specifically about acute high doses during anticipated total sleep deprivation, not daily maintenance during normal life. That is not a small shift. That is the whole frame, rebuilt from the ground up. So what does grounded action actually look like, given everything the evidence does and does not show? It looks like this. If you have a known sleep deprivation event coming, an overnight shift, a cross-continental flight with immediate cognitive demands on arrival, an all-nighter you cannot avoid, the mechanism is most plausible there. The form is standard creatine monohydrate. The doses studied are substantially above the 3 to 5 grams most people take daily. Timing relative to the deprivation period appears to matter, and the honest ceiling on the effect is attenuation of decline, not restoration to full rested performance. You are raising the floor slightly, not rebuilding the ceiling. That distinction matters every time you evaluate a claim about this compound. Drop a comment below telling me which piece of this shifted your thinking the most. Was it the blood-brain barrier and the saturatable transporter, or was it the size of that dose gap between what muscle protocols use and what the sleep deprivation research actually tested? And if you want to go one layer deeper, the next video maps what happens inside your brain's energy system hour by hour across a full 24 hours without sleep, independent of any supplementation at all. Here is the part most people never expect. Some brain regions actually increase their activity in the early hours of sleep deprivation as compensatory mechanisms fire up before the collapse arrives. That failure mode sequence is coming next. Subscribe so you don't miss it. The broader principle underneath all of this is worth carrying forward beyond creatine. Your body's response to any compound is not fixed. It is conditional on the biological state you are already in. Context determines effect. That is a more powerful idea than any single supplement recommendation, because it applies to every claim you will ever encounter about performance, recovery, or cognitive support. Go back to that 3 a.m. image from the beginning, the sand-packed eyes, the sentence you read 3 times, the hand that moved a second too slow. That is not just tiredness. That is your phosphocreatine buffer running thin, your transporter gate controlling the only road into the brain, your prefrontal cortex losing its fuel supply in real time. The scene is the same, but now you can see the machinery underneath it. And that changes everything about how you read the next headline that tells you a supplement reverses brain fog.